right, up next is Dr. Kevin Flanigan, and Kevin is going to talk to us about genetic modifiers.
Thank you. Thanks for the invitation to speak. It's always a pleasure to come to this great conference, and a pleasure always to speak after Stan Nelson, who gives such a great overview of genetics. I was tasked with talking about genetic modifiers, things that change Duchenne muscular dystrophy. So, as Stan pointed out, we lump the dystrophinopathies into Duchenne muscular dystrophy and Becker muscular dystrophy. This might be no better, I think. Is that okay? All right, okay. With sort of the classic definition of Duchenne dystrophy, the loss of ambulation by the age of 12 or so, although this has changed in our modern care with steroids. And Becker muscular dystrophy being the term we use with loss of ambulation sometime after that. For much of the work I'm going to tell you about, we have categorized large cohorts of patients, including a group we call intermediate, as other investigators have done. Those who lose ambulation between 12 and 15 years old, some because they're on steroid treatment, some because of genetic differences, how the protein is made, and so forth. But this leaves the term Becker muscular dystrophy for loss of ambulation after the age of 15. And as Stan pointed out, this goes into people who, there's many types of Becker muscular dystrophy, boys who stop walking at 16 or 17, and men who stop walking at 45 or 60 years old. You've heard all about the reading frame rule, how the size does not correlate well with the phenotype, this the clinical syndrome, and how in-frame deletions are more likely to result in a protein with partial function, and out-of-frame deletions are DMD about 90% of the time. But what accounts for patients who break the reading frame rule? And what accounts for variability just with among patients with DMD? Some lose the ability to walk at 8 years old and some at 12 years old. And that's really what I'm going to talk about today. I'm not sure that this is any better. I can maybe avoid popping. Let's try this. Is that okay? Is that all right for everyone? Sorry, the ringing was getting to my ears there. I apologize. So, I'm going to tell you data that's largely derived from a project we did called the United Dystrophinopathy Project, where we gathered DNA and clinical information from 7 different centers. We gathered historical and prospective data and gathered DNA samples that were tested in the laboratory of Bob Weiss at the University of Utah. And I know some of you have letters from us from this project. We did genotyping for you. And we also have some additional data that was provided to us by patients in an online self-reporting. For this project, we took patients with a clinical diagnosis of DMD or BMD and an excellent family history, or muscle biopsy that showed absent or altered dystrophin expression, or DMD gene mutation testing in previous clinical settings. We took anyone called dystrophinopathy and sort of reanalyzed them. And this work was done beginning in about 2003. We wound up reporting most of the mutations and the phenotypes that went with them in a paper when we described this large cohort of patients. How large? About 1,111 patients with Duchenne and Becker muscular dystrophy manifesting carriers or non-manifesting carriers. And so this is the group that really forms the basis for most of the continued work we're doing now, including our current NIH-funded project. So when we looked at all of the patients in this reading frame rule, we found out, as predicted for DMD, about 90% of the patients follow the reading frame rule. But for patients with Becker muscular dystrophy, actually only about 50% of Becker muscular dystrophy patients have out-of-frame mutations. So we wanted to explain in part why that was. So there's a couple of different ways that one can modify the phenotype. The first is modifications at the Duchenne gene locus itself, at the DMD locus. I'm going to talk about 2 examples of this. Altered mRNA splicing due to what are predicted to be nonsense mutations, but in fact don't function as nonsense mutations. Instead, they cause splicing difficulties. And I'm just going to gloss over another thing, which is altered protein translation initiation. In fact, I won't talk about this, but I'll just point out that there exists in the United States a single nonsense mutation very early in the gene that we would predict would cause severe disease, would cause essentially a very mild phenotype. And we are still continually turning up patients who all are the descendants of some unknown North American pioneer. And I'll talk as this example first about nonsense-associated Becker muscular dystrophy. This is a graph of all of the patients with nonsense mutations out of our 1,100 or so patients. showing the distribution of whether we call them Duchenne or Becker muscular dystrophy. And you can see that all of the patients called Becker muscular dystrophy, essentially all those patients, are clustered in the shaded area. That shaded area represents every exon from Stan's cartoon, where if we took out that single exon, if you skipped that exon, you would still have a continuous reading frame, an open reading frame. So this tells us the nonsense mutation-associated Becker muscular dystrophies are found in this region where there's a zero frame constant, a skippable context, I should say, where you can remove those. How does it do it? Well, what happens is in patients like this, I'm sorry, I'm jumping here a little bit. This happens by a process in which the nonsense mutation doesn't function as a place where translation stops, but instead functions as a way to alter the signals that are encoded in the gene to splice. These are the exon splice regulatory signals. Those signals that are within the gene that say, hey, I'm an exon, splice me here. That's what we have. Now this is a very busy cartoon from a French, it's impossible to read in the back. I should finish my sentence. It's not because it's French. It's a very busy cartoon from a French group that described this back in 2006. And we think of skipping of exons or splicing exons as a fairly simple process, but there's a whole bunch of proteins that have to be recruited to the exon in order to form what's called the spliceosome, the machinery that actually snips the introns out and puts them together. And if you look right down here, a white dot down here, the group of Dystrophy in French described a patient in whom what was predicted to be a nonsense mutation, instead, we made what's called an exon splice suppressor site. It caused the recruitment of proteins that interfered with the whole assembly of this splicing machinery. So that spliceosome inhibition is a mechanism then where this exon doesn't get recognized as an exon. It gets excluded totally from the transcript. And in fact, this exists, it's well defined. So in those patients, I'm not sure if I can go back actually. That example I showed you for one example is exon 31. And in fact, all 5 patients in our database that had nonsense mutations with an exon 31, in fact, were Becker rather than a Duchenne phenotype, at least in our sample. So besides modifiers at the DMD locus, so that's one way to think about it. How about other genes elsewhere that modify? As you saw, Duchenne dystrophy is a disease not only where there's a problem with dystrophin, but there's fibrosis of muscle, there's fatty replacement of muscle, there's inefficient, there's a lot of regeneration of muscle. All of these are influenced by genes. So can genes elsewhere influence disease severity? So that's the second part of this talk. And in fact, there's a couple of examples of this. The group of Dr. Pecoraro and Eric Kaufman published data on a gene called SPP1, which encodes osteopontin. Osteopontin is a cytokine. It's involved with immune cell migration and survival, and it's implicated in fibrosis and the creation of scarring in muscle through the TGF-beta pathway. And their group showed that polymorphism, that's just a variation that exists in the population, that this variation in the SPP1 gene is associated with decreased strength in steroid-treated patients with Duchenne muscular dystrophy. And there's a difference in the disease severity as described by loss of ambulation, whether you have one type of this polymorphism or another. The second one is, I'll show you data from our own group. Actually, it's derived from work from Dr. Beth McNally at the University of Chicago. Dr. McNally studied a mouse model. You've seen a cartoon version of this already, binding to between actin and dystroglycan in the membrane. Well, Dr. McNally studied a mouse called with a mutation in gamma-sarcoglycan. This is a model of limb-girdle muscular dystrophy type 2C, but also it has similar muscular dystrophy. And in that mouse model, she did a bunch of measurements of things relevant to muscle disease. How leaky the muscle membrane is, how much fibrosis is there. So you can measure leaky muscle membrane by how much of a dye muscle fibers take up. You can measure fibrosis by the signal you get when you stain with hydroxyproline. And she did a test of two strains of this mouse, 2 strains of the gamma-sarcoglycan mouse. One that had severe features and one that had mild features. And after many crosses and many, looking at over 270 animals, she found that there were signals at 2 places in mouse chromosomes. Well, there were signals for each of these traits in the mouse. This signal you can see is the statistical estimate of a modifier being removed there. You can see that peak on chromosome 7, and this is the modifier for membrane leak. And she saw a similar signal for a modifier of fibrosis, and these overlapped each other, giving good evidence that there was a gene modifier within this region. And sure enough, there is a very interesting modifier in that region, a gene called latent TGF binding protein 4, LTBP4. So it's not terribly important to remember, except you note that it's in the TGF beta pathway. Again, very important for fibrosis and inflammation. And it's actually a member of a whole group, a superfamily of proteins. They have a particular role. What they do is they regulate the availability of TGF beta. This is a very busy cartoon. A TGF beta protein here that gets sequestered by another latent protein, and this gets sequestered in the whole complex. There it sits until it's ready to do its work. And when signaling occurs, it gets released. So Dr. McNally showed the difference between her 2 mouse strains was a difference of an insertion of an area that's called the proline-rich region. They differed by the presence or absence of a chunk of peptides in there. And in fact, this is right at the site. This is right at the site where cleavage of this latent protein occurs. Proteolysis is what we call cleavage. And that proteolysis is a part of the process by which TGF beta then gets released. It goes to its receptor, and when it goes to its receptor, then signaling can occur, and all the downstream effects, fibrosis, inflammation can occur. So it all fit together nicely with what she found in mouse. And I'll just point that this region is quite variable, including between rats and humans and other animals. So it turns out in humans there's variation in this gene as well. This is, I'll show you here, I'll call your attention to these letters V, T, 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 which are symbols for individual amino acids. There's a version of this gene in the population that goes V, T, T, T at 4 sites across there. And there's a version where there are substitutions at each of those sites. There's different amino acids at each of those sites, I, A, M. So those 2 versions are what are generally in the population, VTTT at a frequency of about 48%, IAAM at a frequency of about 37%, but they go together, they track together. So I'm going to just show you examples of one or the other in a minute here as we look at our population. But we said we wanted to know, like in mice, can LTBP4 influence the phenotype in human patients' disease? So we use loss of ambulation. You as parents know we can often define loss of ambulation to the nearest half year. Someone stopped walking, let's say, at the beginning of the school year, the end of school year, during the summer. We can often pick on that date. So we could use that date as a marker that was relatively reliable. And we wanted to make an unbiased approach. If LTBP4 had a beneficial impact, well, boys we called Becker, we didn't want to make any decisions about what we called the patient, whether we called them Duchenne or Becker. We took all patients who stopped walking before the age of 20, and out of those boys, regardless of what phenotype we called them, we included them in our analysis. And we had 254 subjects in this population. 244 of those were cataloged as Duchenne, a couple of them as Becker muscular dystrophy, and 8 of them as this intermediate phenotype. And when we looked at these, we found that IAAM haplotype predicts prolonged ability to walk in boys with Duchenne dystrophy. So these curves are seen in these. If you haven't seen scientific talks, there's often curves like this that show the percentage of people that can do whatever function it is over time. And we can see here as it goes down, the percentage of boys walking marked by their is higher for each patient carrying the red line, which is the protective version of this haplotype. So there's a significant difference in the ability to walk if you have the protective version. And I'll briefly say, this would be hard to read from the back, but the prolonged ambulation in this group was not explained by the type of DMD mutation, whether it's in or out of frame. It was explained not by whether it was a truncating mutation or not. but was explained by the LTBP4 phenotype. And this seemed a particularly vigorous signal when we looked at steroid use. Steroid use prolongs this, makes it actually extends this, makes this curve out farther. And when I'm going to skip over this one to say when we look at steroid use for these variants, we can see highly statistically significant numbers. For all patients, it's significant. For the steroid use variants, it's really quite significant. And what we call a p-value that is strongly argues for a relationship. So the mean age of ambulatory loss for IAM versus other haplotypes in steroid-treated patients, it means a difference of about 1.8 years. In steroid-naive patients, about 1.4 years difference in walking, whether you have this protective version or not. And just one thing to follow, to close the story, is when we look at fibroblasts from patients with this, the downstream signal of activity of TGF-beta goes along with what we would expect. So that there's less TGF-beta signaling marked by the darkness of these upper bands. Phosphorylated SMAD is what we're looking at here. And it's more, there's less of that signal in the groups with a protective haplotype. So altogether, this tells us that LTBP4 is associated with decreased TGF-beta signaling. It's independent of the primary mutation. It may act through glucocorticoid. There's an effect both on steroid-naive and steroid-treated patients, but the magnitude of the effect is greater in the setting of steroid treatment. So we should consider this when we analyze data from clinical trials. That is, we have to interpret whether or not boys have that phenotype, or have that genotype or not when interpreting data. And I should point out this information provides a new pathway for therapy. I won't speak to Dr. McNally's data, but I know that Dr. McNally, in fact, is looking at development of therapeutics modulating specifically this protein's function. So I'll close to say, we have this big database of patients, 1,100 DNA samples, a lot of information. Many people in this room participated in the United Dystrophinopathy project in the past. And we're funded now by the NIH to go back and recontact the families who participated in this. So you may receive a call from us if you have participated, where we're going to go back and seek to gather information related to certain outcomes: age of wheelchair use, age of diagnosis of cardiomyopathy, age of BiPAP use, survival. And we've already looked, we've got about 2.5 million variations in genes across the entire genome. Already genotypes in hand for about 800 of our patients, 800 of our subjects, and now we're going to be looking at those in order to identify new genetic modifiers and new pathways. So I'll close with that and just point out that much of the data that I presented to you today was data in collaboration with Beth McNally and her team, and Bob Weiss, my longtime colleague at the University of Utah. Thank you.